morning. We're starting a little late. It's my great pleasure to welcome everyone here to the Alzheimer's Drug Discovery Foundation's November Symposium. 18 years ago, my brother Leonard and I founded ADDF for the simple reason no one was helping fast-track drugs. These drugs to treat Alzheimer's and other causes of dementia. Today, you will hear just how far we have come in this effort. And I can tell you, the news is exciting. The panel will be led by ADDF's Executive Director, Howard Phillott, and he will bring you up to date on the progress we are making. It's thanks to his hard work and the ADDF staff and the scientists that we fund across the globe that we are closer than ever to having effective drugs that will fight these diseases. For many of us in this room whose families have been touched by these devastating diseases, and that includes Leonard and myself, these drugs can come soon enough. After the presentation and questions, I hope you'll all join us for lunch when we will honor Donald Newhouse for his tremendous leadership and partnership in supporting the ADDF and more specifically, the treatment for a rare form of dementia. Donald is here with his daughter, Kathy. Donald and Cassie, please stand up so all of us can give you a round of applause. Thank you, Donald, for your wonderful help. I will have more to say during lunch, but right now, it's my great pleasure to introduce Dr. Howard Phillott and the panelists. Howard, it's all yours. Thank you.
Thank you, Ronald. And I want to thank everyone for coming today. We really appreciate your support and your interest, and I'm sure your personal involvement in Alzheimer's disease through family and friends. Today we have a great panel of speakers. I'll be introducing them. And we're going to do something a little different. We're going to tell you about 4 different programs that ADDF is supporting around the world to give you an idea in depth about what it takes to develop a drug for Alzheimer's disease. To begin, I just want to review for a second our mission. We are the only foundation in the world that is solely dedicated to the discovery and development of new drugs for Alzheimer's disease and related dementias, and the one we'll be talking about today that Don Newhouse and his family are helping us to support is frontotemporal degeneration, which is related to Alzheimer's disease and the pathology, and it's a great model for us to work together in partnership. So this is really a very different field of science to be developing drugs. It's mostly chemistry, basic research is biology, and so we really help biologists and researchers at universities and small biotechs around the world to accelerate the development of their drugs by bringing expertise and resources to their programs. As I mentioned, we're going to be talking about different dementias. Alzheimer's is the most common cause of dementia, dementia being a syndrome of people losing their minds, basically, a clinical syndrome that we define. And Alzheimer's is about 70%, but as you'll see in the list here, vascular dementia is quite common, especially in the elderly. We all basically develop some form of vascular disease related to hypertension and other illnesses like diabetes and atherosclerosis. And you're going to hear today from Sandra Black about how we're trying to approach how the vascular aspects of the disease affect the brain, and in many cases, patients have both Alzheimer's and vascular dementia, and they tend to progress more rapidly. And so you see that listed there as the mixed form of dementia, which is very common in clinical practice. Lewy body, we're not going to talk about as a program today. We are supporting research. That type of dementia, probably in pathology, affects about 35% of people who come to autopsy. We don't have a biomarker like we do now for Alzheimer's disease, but we're developing biomarkers like PET scans and other tests, spinal fluid tests, to detect these people. And it's related to Parkinson's disease. So there's sort of a continuum between Parkinson's disease, dementia, Lewy body dementia related to Parkinson's and Alzheimer's in the way that people present. And then frontotemporal dementia, which you see here, which tends to affect people earlier in life and has a different presentation. And we're going to hear from Dr. Huey about what frontotemporal dementia is and what we're trying to do. The thing is that all of these dementias are neurodegenerative diseases. They're all chronic, progressive, and really uniformly fatal neurodegenerative diseases. And while we don't understand why their pathologies are different, Lewy body, vascular, in Alzheimer's we get plaques, in frontotemporal dementia and Alzheimer's we get tangles. While we don't fully understand why these diseases are expressed clinically somewhat differently and have different pathology, we know that aging is the leading risk factor for all of these illnesses, and that there are certain common multiple disease mechanisms related to aging, which we can learn from in the different dementias and apply them to the development of new drugs. So we're going to hear today from Carlos Buesa from Oryzon in Barcelona, a biotech company we're supporting, on how epigenetics can be modified to develop drugs for Alzheimer's disease. Neuroinflammation is very important. Inflammation plays a critical role in aging and critical role in Alzheimer's disease. Neuroprotection, a final common pathway by which we might be able to protect neurons from dying and being injured, regardless of what the form of injury is. So sort of drugs that could ultimately affect and be useful for all forms of dementia. And mitochondrial dysfunction, those are the energy powerhouses of the cell, and things like insulin resistance, poor glucose utilization in the brain, and energy failure in the brain is very important. And unfortunately, we won't hear about that today. But just to give you an idea of the broad but rather focused way that we're identifying specific target areas in our strategic portfolio management of our science program and focusing our efforts on very specific mechanisms that are common to these neurodegenerative diseases to develop drugs that will treat them and ultimately, hopefully, prevent them. So let me introduce our first speaker. Dr. Sandra Black is a professor at the University of Toronto. She's the Brill Chair in Neurology, a very well-known international clinical trialist. She's also the executive director of the Toronto Dementia Research Alliance, which is bringing together multiple entities in Toronto to accelerate clinical trials. ADDF is funding her to develop vascular targets to look at antihypertensive therapies that can be used, repurposed, for the treatment and prevention of Alzheimer's disease. And she's going to tell us more about that. So welcome, Sandra.
Well, I'm delighted to be here. It's a great privilege. And just so you know, I think the traffic in Toronto is actually worse than New York, but maybe not on Fifth Avenue. So I have the privilege of telling you about one of the projects that we're involved with ADDF, and it has to do with blood pressure. I think everybody in this room knows that high blood pressure is not good for your body or your brain. It's related to a heart attack, stroke, kidney disease, eye problems, but I don't know if you are all aware that it also is a risk factor for Alzheimer's disease, as well as other vascular problems, smoking, physical inactivity, high cholesterol, diabetes. So hypertension has now been studied, and it turns out to be a very important factor in brain health and aging. And so control of it, particularly in midlife, is very important. By 75, which is the rough onset time of Alzheimer's disease, about 64% of people have hypertension, and so it really is very important to treat it and treat it well. So there are studies that have shown in autopsies, for example, the Honolulu aging study, where they followed men who had uncontrolled hypertension in middle age 40 years later when they donated their brains to science, and they found that there was a correlation with the plaques and tangles of Alzheimer's disease after you corrected for all of the risk factors. There was a direct correlation. And I'm going to show you a slide, if we can have the first slide and maybe the pointer. That is taking advantage of fast forward to modern imaging techniques where we can do brain scans and actually look at the thickness of the cortex in people who are normal elderly versus individuals with Alzheimer's disease. And you also may be surprised that although in the end stage Alzheimer's seems to shrink the whole brain, it actually starts off affecting particular functional systems. So these are the peak areas in Alzheimer's disease. This is showing a side view of the brain, the middle hemisphere from the inside out, the lateral surface and underneath, showing peak areas that show thinning in almost everybody as they develop Alzheimer's disease. It's part of a functional system that we're beginning to understand, functional systems that we're beginning to understand are important in how it manifests. Now look at what happens when you have untreated or people with high blood pressure. Actually, these were people who were treated, but they were having trouble controlling their blood pressure, and they just followed them for a few years. And guess where they showed thinning? They didn't have to be autopsied to see this. These are people who are alive, and they're having simple MRI scans. And it's very interesting that exactly the same areas, many of the same areas overlap. So there seems to be some coalescence of these risk factors in certain key areas in the brain, which is perhaps why the commonest substrate of dementia at autopsy in population studies is actually a combination of Alzheimer's and vascular disease. Hence, treating blood pressure is very important, and we have many, many medications, but some may be better than others for the brain as well as for the body. So what we have chosen to do is a comparative effectiveness trial comparing two classes of drugs, but we've chosen the best in class, that affect the renin, it's the kidney system, and control blood pressure. They're good for the heart. They have protection of the blood vessels, and they also bring down blood pressure. And in the brain, there's a whole similar enzyme system that can be affected by these medications. So it turns out that one set, they're called sartans, they're the angiotensin receptor blockers, as opposed to the angiotensin receptor inhibitors. You may know the term ACE inhibitors or ARBs, or sartans for ARBs. It turns out that the sartans may have additional beneficial effects. They actually inhibit the breakdown of amyloid, so they're protective in that sense. They have effects on another enzyme or another receptor that improves glucose uptake and energetic performance. They have anti-inflammatory, anti-atherosclerotic effects. Therefore, we are taking individuals with Alzheimer's disease in the mild to moderate stage, and we are randomizing them to a drug called perindopril, which is an ACE inhibitor, or a drug called telmisartan. And we're going to follow them over 1 year. We're studying them in many ways, but we're going to follow them over 1 year. And our prediction, our hope, is that we can show that the brain is going to shrink less in the people on telmisartan because of its neuroprotective effects. These have been shown in a big veterans study where they had individuals not randomized, but some were put on an ACE inhibitor and some were put on sartans. And it looked like the people on sartan were less likely to develop dementia and to decline cognitively and be hospitalized. So there's another study where at autopsy, individuals who were on sartans, who were hypertensive Alzheimer patients, showed less pathology and less a slower slope of decline. So we have epidemiological evidence, but now we actually want to do it in a randomized prospective way, and our hope is that we will be able to maybe give advice for which may be the best drugs to use in people with Alzheimer's or just elderly individuals who may be at risk of Alzheimer's disease. So that's the plan, and we're very grateful to have this opportunity. This is called repurposing. You can fast track treatment decisions with repurposing drugs because they shave off about 10 years in the development. And these two drug classes are extremely well understood and very well known from the point of view of their antihypertensive effects. What's intriguing is more and more we're finding out that they have differential effects in the brain. And so what is good for the heart may not be as good for the brain as another alternative. And so what we're expecting and hoping is that the sartan drugs are going to show us a beneficial effect that may translate very quickly into better management for blood pressure, middle age, later age, and by the way, I don't have stocks in any of these companies. In fact, one of them is generic now anyway, so that they're really, it's of no interest to the pharmaceutical companies, unfortunately, and you're going to hear about some more repurposing efforts where drugs that are already available have to be reconceived. So in fact, telmisartan to us is a cognitive enhancing agent, a neuroprotective agent, which also happens to be an antihypertensive agent. Thank you.
Running clinical trials for Alzheimer's disease that you're facing?
Well, actually, what's very interesting is we have to compete also with pharma trials with new drugs, and we're very involved in that as well. But we've run into some, I would say, cultural conservatism among some of our tertiary clinic physicians. They can be geriatric psychiatrists, they can be cognitive or behavioral neurologists, or they can be geriatricians. And they've got this kind of fixation that these are antihypertensive drugs, and they're having trouble switching to think of them as cognitive enhancing and neuroprotective drugs. And they're also not used to managing them. So one of the things that's been challenging to us is getting them to be willing to participate in the trial. And we've actually, now we're turning more and more to family practice, primary care. And we're taking advantage of large populations in Toronto with electronic patient records where we can find out the appropriate patients. And so we thought that would be the next phase, that we would go to family practice after we had shown the proof of concept. Now it looks like we're going to get family doctors referring at least the patients and being involved right from the get-go. So that's been a little bit of a surprise to me, but it's also that sometimes it's not as sexy as a new drug, having something that everybody knows. And yet these are very powerful medications. Both the ones we're using are actually helpful in the brain compared to other antihypertensives, but the telmisartan drug, telmisartan, has all these different effects, which all end up being positive. So that's what we're going to do.
So could you imagine a new class of agents that would be sartans that might be derivatives of the current sartans and would be used not just in people who have hypertension, but actually we're actually funding a clinical trial of sartans just for people who are at risk or have early Alzheimer's disease, so that we can actually test in the clinic.
That's a great idea. I mean, I think the other advantage of these drugs is that they're also drugs that affect the vascular system, which is very important in the development of Alzheimer's disease. Alzheimer's disease is also a disease that affects the vessels. I often shock some audiences and say, do you include Alzheimer's disease in your risk factor for stroke? Because Alzheimer's can cause hemorrhagic stroke, because amyloid goes into the vessels as well. So it actually protects the vessels. And it would be nice to maybe try to derive what the key essence is, but I think part of the reason that these drugs work so well is that they hit so many targets.
Right.
Thank you.
And thank you, Sandra. We'll have time. We'll have time for questions from the audience. I think the other thing that after the speakers, the other thing about the sartans and these repurposing drugs is that, let's say the trial is completed in 2 or 3 years and it's positive. As a physician, I would be able to prescribe that drug for my patients the day that the drug becomes shown to be effective. So we wouldn't have to wait for that 10 to 12 years of a long process of FDA approval. Our next panelist is Carlos Buesa. He's the founder of a biotech company in Barcelona that we're funding called Oryzon, and we've been very proud of the progress of the company, and we've really enjoyed working with them. They just went public on the Spanish exchange in December. They're a cancer company. They have a hundreds of millions of dollars relationship with Roche on their cancer target, and we've helped them to develop their Alzheimer's program. And what's really, really fascinating about the Oryzon program is that the target in that case is epigenetics, and Carlos is going to tell us what epigenetics is, but it's not the same your grandfather's genetics. It's a very different type of genetics that really regulates how the genes work. And so Carlos, we've helped to move from preclinical research in animals, and now they're doing a clinical trial of their lead candidate drug for Alzheimer's disease. So we're very proud to have Carlos here today. Thank you, Carlos. Thanks for coming from Barcelona.
Thank you, Howard. It's a real pleasure and honor to be here supporting the ADDF and to be grateful for your support as well. Well, I'm going to talk you about what is first epigenetics, which is a weird name, so everybody knows about genetics, which is the DNA from mommy and daddy, and grandpa and grandma. But what is epigenetics? So I don't know if I can have the slide. I'm controlling the slide. I'm sorry. So what is epigenetics? I'm going to try to show you an example to exemplify how epigenetic works. So we know that we have our genomes, our genes, and we know that this heritage that we have from our families, from our ancestors, is basically not changing unless we have a mutation. So what we can say is that epigenetics is a way that our cells use to regulate how this genome, how these chromosomes work every single day of in every single cell, in every single day of our lives, to adaptate ourselves to the normal living circumstances. So it's a kind of interface between the outer world and the inner world. So it could be like in a old-timer telephone switchboard, when the wires were there, but we were needing an operator to connect lines and to unplug lines. And by plugging and unplugging, by this mechanism, there are whole sets of the chromosomes, certain genes which can be turned on or turned off. So what it happens is that the operators of this microscopic genomic switchboard are not these ladies, but they are proteins, they are enzymes, which are basically plugging or unplugging small molecular edits on the chromosome, on the proteins that serve as a scaffold to the DNA. And by doing so is sufficient, like in the movies when there is an actor which is basically finding a treasure, and just by moving a small piece of the wall, the whole wall moves or collapses, is the same at the molecular level, so we can get activated whole regions of the genome, and this is happening every single day. Unfortunately, when something goes wrong, then we can get a mess. And this mess is not going to be like in a movie comedy, is going to be probably a disease. We know today that Alzheimer's disease, neurodegenerative disorders are also having a strong epigenetic component. And there have been a number of very beautiful papers published by the different academicians. And one which I like very much because it's a very nice image is the report of two siblings, two old ladies, which were genetically identical, homozygous, and they were harboring the same mutation for predisposing them to develop Parkinson's disease. In spite of being genetically identical, one of the sisters was developing Parkinson's 20 years before than the other. So this is illustrating how this wiring, how this epigenetic modulation of our genes can be, at the end of the day, fundamental on the progression of some diseases. Okay, so this is epigenetics, and this is epigenetics and neurodegeneration. So why are we working on epigenetics? Well, we were starting in Barcelona in 2007-8 in a program that was a genomic survey on autopsied brains from elder people that had died without clinical manifestations of any neurological disorder. But when the pathologists were looking under the microscope to these brains, they were seeing already the initial clinical hallmarks, the initial lesions on the brain for Parkinson's disease, dementia of the Lewy body, and Alzheimer's disease. So we were interested at that time on the genomic reasons of these chains, and we end up in LSD1. LSD1, which is a funny name, but is only referring to lysine-specific demethylase, is an epigenetic modulator, is an unplugger. It's basically removing methyl groups from the genomes, and by doing so, is regulating how the genes of our neurons work. Okay, so at that time we were starting to work with LSD1, and it took us a while to develop a first molecule that was good enough to be developed in preclinical studies. We were studying this molecule in a mice model, which is basically a SAMP-A model, a model which was developed in Japan and by the University of Kyoto. And these mice, they basically experience a very accelerated aging. So they live faster and shorter. And by doing so, at the month 4 or 5, these animals really display strong memory impairments. So when we were treating those animals with our drugs, we were seeing that we were able to restore basically the cognition of those animals. So that was really giving us lots of enthusiasm, and at that moment we were starting to be helped by the Alzheimer's Drug Discovery Foundation, which has been key on supporting our company in several times on the evolution of this process. So we have been working for 3-4 years on these animal models and in other animal models to really assess what's happening inside the brain when we treat with this inhibitor of this unplugger, of this epigenetic modulator. And what we know now is that by inhibiting LSD1, in those animal models, what we provoke in the regions of the brain where the memories are created, we provoke an increase of the gene expression of neuronal survival, an increase of the gene expression of neuronal plasticity, the way that the neurons talk to each other, and we provoke a decrease of a set of neuroinflammatory genes. So neuroinflammation has been also said or suggested as one of the causes or accelerative processes in the neurodegenerative disorders. So this dual combination, these neuroprotective effects plus these anti-neuroinflammatory effects, we believe that they are on the basis of the phenotype of the effects that we see on those animals. So we got the crucial help of the ADDF to do the final preclinical regulatory talks, and we are now very happily ending up the phase 1 study, a phase 1 study which is being done in healthy volunteers to assess safety and tolerability. So we expect to be ready to start a phase 2 in Alzheimer's disease in the second part of next year, which we expect that is going to be sufficiently clear to show efficacy eventually efficacy in humans. Thank you very much.
Thank you. Just a couple of questions for Carlos. One is, so based on the many mechanisms of aging, I mean, the current drugs for Alzheimer's that are in advanced clinical trials try to remove the amyloid, but that's just one piece. We don't even know if it's going to work. So could you comment, because you mentioned, I think, or maybe just a comment that there's so many different ways that the neurons get injured, and it seems to me like epigenetics is a way to address the common pathways, the many common pathways that cause neuronal injury. So it could be a drug for the disease in the sense that it addresses so many different mechanisms that it could be very efficacious across, and even preventative, I would say.
I mean, yes. Epigenetics is a mechanism which is very, we say the scientists, upstream. So that means that it's very high on the level of regulation. So there are many effects or many pathways that we can address by refixing or setting up correctly what is going wrong of this superior mechanism. We know that A-beta is playing a role, and A-beta is probably playing a strong role in familiar versions of Alzheimer, the amyloid deposition. But the etiology of the causes of the sporadic Alzheimer are much more unknown. So we probably are facing, when we talk about Alzheimer, we probably are facing a number of diseases, which one can be caused by oxidative stress, another can be caused by problems on getting rid of the mixed folder proteins. I mean, there are several reasons why a cell can be sad, can get blue, and can die at the end of the day. What happens in the brain is that at the end of the day, when the neuron dies, we get the same symptom. But the ultimate reason why the neuron was dying can be different. So if we go to the fundamental reasons why the neuron is getting blue, we can eventually address several of these combined diseases.
Thank you. Thank you. Okay, thank you. Our next speaker is Dr. Michela Gallagher. She's one of the leading neuroscientists in our society. She's been at it for a long time, and she did a lot of basic research over many years, and then came across a very interesting drug that she's going to tell us about, which is, again, another repurposed drug done in a different sort of way than what Sandra Black is doing. Michela spun a biotech company out of Johns Hopkins, where she's a professor, called Aging Bio, and we've been funding her for several years now, since 2010 at Aging Bio, to develop this drug, which, exciting, is now in phase 3, which is the last, we're hoping to go into phase 3. She's fundraising for phase 3 right now, which is the last phase of clinical development before a drug is proposed to the FDA for approval. So, and it's in another part of Alzheimer's called mild cognitive impairment. She'll be treating that, for which we have no drugs at this time.
Yeah, thanks a lot. I'm here to sing the praises of this foundation. It's just incredible. We definitely would not be where we are without it. So as many of you know, from the initial diagnosis of Alzheimer's dementia in a patient, there is a very slow, maybe I could get my slide.
Yeah, just press the green button.
This one, okay. Yeah, so from the first clinical diagnosis of dementia, there is a very lengthy progressive decline in a patient. In an otherwise healthy person, this can last many, many years, as it did with my mother, who from diagnosis took this awful journey to her death 12 years after the initial diagnosis. Mild cognitive impairment, which I'd like to introduce you to today if you're not familiar with it, it refers to an earlier phase of the disease, and it's also very lengthy. We've learned a great deal about this condition of mild cognitive impairment in recent years, and it's now centered this condition in patients, which can be diagnosed clinically, as a target for prevention of dementia, okay? So the concept here is if we could stop the progression of Alzheimer's disease in this early phase, that would be an amazing achievement. As the name identifying this condition suggests, mild cognitive impairment, there are symptoms that differentiate this condition from normal aging. Primarily, that memory problems are worse than you would expect for a person's age, and they are slowly getting worse than that. But mild cognitive impairment is defined as a condition where people still have intact activities of daily living. They're really functioning. So that if you could stop at this point, you would have a person with some disability, but the burden of care would be relatively minimal, and then we could work on those symptoms, okay? And as Howard mentioned, currently there is no treatment, symptomatic or to slow progression for this condition of mild cognitive impairment. Now, even if you couldn't stop it entirely, slowing progression during this phase could be of major benefit. So say a person who this might be a decade-long phase, if you could extend that 4 years, you would have 4 years of functioning and quality of life that you wouldn't otherwise have. And because none of us live forever, if you could extend mild cognitive impairment, that diagnosis of dementia, by 4 years, very good, solid public health studies have demonstrated it would decrease the prevalence of patients with dementia by 30 to 40%, which is amazing. So that is really a mission that is worthwhile, and the mission I'll talk about with the drug development program that ADDF has sponsored. The one thing I just want to point to up there, these are two little pictures of the world today and in 2050. And the dark blue areas are areas where the population is already top-heavy. There's lots of aging in Japan and Western Europe. By 2050, that condition will affect all the blue areas on the globe on the other side. And it's predicted that, dementia, the prevalence of dementia may increase three times by 2050. So preventing dementia is a very, very worthwhile thing to try to do now. Now, the work that we are currently doing is highlighted on the bottom. There is a clinical trial. We've had our last meeting with the FDA before enrollment. We've checked all the boxes. We're ready to go. This trial is sponsored by a company I founded out of Johns Hopkins called Aging Bio. And the Hope for MCI is the name of the trial, and there is a website that gives information about it currently. And the other thing about this trial, and I'll talk about repositioning, our drug is not exactly a repositioning drug. It's a little bit more complicated than that. But this drug has received support from the NIH as partial support to pay for the costs of conducting the trial. So I said that we've learned a lot about mild cognitive impairment in recent years. And one of the things we've learned is that by using imaging methods that actually show brain function in people while they're performing memory tasks, it was discovered that these areas in the memory system of the brain are very overactive. They're activating like crazy when people are, that activity is being measured. Initially, it was sort of thought, well, these people have memory problems, maybe that overactivity is compensatory. That memory system is working harder to help support memory. But the discoveries that I made in my basic research program at Hopkins led me to believe that the overactivity was part of the problem. And we actually did a research clinical trial sponsored by the NIH based on screening many medications that we thought might be able to bring that overactivity down. And on the basis of the results in animals, we tested this one medication that worked in humans. And it did bring the overactivity down in functional imaging. And then the people's memory function that depends on that system improved. So we've already got a little symptomatic signature in these patients with amnestic mild cognitive impairment. Our ambition is greater than that because the scientific evidence about the pathology of Alzheimer's disease, which is really aggregating where the first degeneration in the brain occurs in the memory system, is driven by neural activity. So that both the amyloid and these tau pathways, in fact, the tau starts there and it spreads along connectional systems driven by neural activity. So our great hope is that if we can continue to put people on this drug over a period of time, that spread, which spreads the damage, the pathology, and accumulates all the cognitive and clinical deficits, could be at least slowed down, So just a couple of comments, if I have a minute or two more, about repurposing. So this is a drug that has been in use, that we are using, that has, the chemical has been in use. It's called an atypical anti-epileptic, so used as an adjunct therapy in patients with certain forms of seizures. This drug has been used in all sorts of patient populations, in children for chronic durations, and it's safe. Not only that, when the drug is used to treat epilepsy, it is given at 10 to 15 times higher doses than are needed to control this overactivity in amnestic MCI. And one of the things that has been amazing, puzzling, and it would take much more time than I have here to explain, is that actually those high doses that work in epilepsy don't work in this condition. You need to use the drug in a therapeutic window to bring down this overactivity because there's all sorts of inhibitory, excitatory balances, and we get it just right in this therapeutic window. The key role of the Alzheimer's Drug Discovery Foundation was to help us in formulating a version of this drug at the doses we need that will produce that therapeutic window with a pill taken once in the morning, okay? And we had to run the experiments by dosing people all along the way to keep the therapeutic window correct. So we now have the pill. The FDA has approved the pill. They've approved absolutely everything we're doing in the phase 3 trial, and we hope to launch it Q1, Q2 in 2017. Thank you.
Thank you. Just a couple of comments before we go forward. The name of the drug is levetiracetam. The brand name is Keppra, but you can't just take the Keppra drug, as Michela mentioned, and chop it down and make a small dose of it. It just doesn't work that way. So part of the patent protection around this kind of reformulation and repurposing of this epilepsy drug is the strategy to have this lower dose. And I think the other thing that Michela is an icon for, as having been a professor of neuroscience at Johns Hopkins for many years, and is that she's now become a biotech entrepreneur as the CEO of this company and has learned a great deal about raising money from investors and running a company and running clinical trials. And we're very proud of our ability to help her to transition that role, although I imagine at some point, once the drug is approved, she might go back. Would you ever go back to the university, or are you staying in the company? Doing both. She's doing both.
Yes, I love research.
Thank you. Okay, our last speaker is Dr. Edward Huey, and he is from Columbia University. And he's actually working on frontotemporal degeneration. And of course, we're honoring Donald Newhouse today for our partnership with the Association for Frontotemporal Degeneration, which we've been doing for over 10 years now. So we've had an interest in this illness. It teaches us a lot about Alzheimer's disease. It's a relatively uncommon form, but it's a pure form of tangles in the brain in some cases. Dr. Huey is using an old drug called lithium for the treatment of symptoms in FTD, and we hope that this will also at some point in the future translate into our methods for treating the same sort of symptoms of agitation in Alzheimer's disease, and might even be neuroprotective. So let me introduce Dr. Huey.
Thank you very much, and thank you for inviting me. So, you know, you can see from my bio, I'm a relatively recent, my connection with ADDF is relatively recent, and the other foundation, the other association that is funding this research program is the Association for Frontotemporal Degeneration, which I've been working with for a little bit longer, and also National Institutes of Health. So, you know, I've been relatively recently exposed to ADDF, and before I kind of start in on talking about my program, just want to say, you know, how impressed I've been with both foundations, well, the AFTD and ADDF, in their, you know, real focus on bringing treatments to patients, which is, I think, a focus that can be lacking in NIH and some of the other funding agencies, and, you know, the smart science and the practical science. So, you know, I've been very impressed and very happy with my association with both of these foundations. So as the speaker up here who is focused on non-Alzheimer dementias, you know, I think let me talk initially a little bit about why we should all care a lot about these non-Alzheimer dementias, even those of us who are interested primarily in Alzheimer's disease. So Alzheimer's disease, as Dr. Phillip showed you, is, you know, there's no doubt it is the most common type of dementia, where dementia is a brain disorder that causes progressive trouble in managing your life. But you know, as you saw from his slide, if you add up all of the other types of dementia, all of those other pieces of the pie, it's actually a pretty big chunk of the pie that is accounted for by these non-Alzheimer's dementias. I have focused especially on frontotemporal dementia, which is probably about 10% of dementias, and also, as Dr. Phillip said, it's different from these other dementias in a couple ways. One of which is that it tends to affect people younger, so it's a disease usually of 50s and 60s. And it also presents really not with memory problems. Usually my patients' memories are fine or relatively preserved. It presents more with changes in behavior, personality, and or language. So, you know, reasons why even those of us interested in Alzheimer's should be interested in these non-Alzheimer dementias. One is that I think, as I've said, it's more common than people might think. So it actually accounts for a significant chunk of overall dementia. The second is, I think the non-Alzheimer dementias are underdiagnosed and much more often misdiagnosed than Alzheimer's disease. And I think there's an idea, both in often scientific and lay worlds, that dementia is memory, and that all dementias are a memory problem. But those of us who work with frontotemporal dementia, other non-Alzheimer's dementias, know that you can have dementia without really having memory affected. So these patients often get missed. And my patients, the majority of them are misdiagnosed with often a psychiatric illness. People say, well, their personality's changing, they're depressed, they're going through a midlife crisis, and it's, in my experience, very upsetting to the families to go through the process of it being misdiagnosed for usually often several years. The third reason why I think it's very important, even if you're focused on Alzheimer's, Dr. Phillip also mentioned, is that these non-Alzheimer dementias, and especially frontotemporal dementia, are really excellent model systems. So, something you probably don't know, I'm sure you've heard tau is a very important molecule for several different types of dementia, but especially for Alzheimer's disease. It's the basis of the tangles that make the plaques and the tangles of Alzheimer's disease. But what you may not know is that there are patients out there with tau mutations, so they have mutations that affect the gene that codes for this protein, but those patients don't get Alzheimer's disease. They get frontotemporal dementia for reasons we don't really understand. So I follow several families with tau mutations. And the other issue that has come up with the other speakers is that Alzheimer's disease is complex. It has many different aspects. It's happening usually in older people. My patients with tau mutations are really getting a very isolated, what we call tauopathy, so deposition of tau protein in a relatively young population. Again, they're usually in their 50s and 60s. So our group and other groups have been very interested in, again, using this as a kind of proof of concept model population. So if you have an imaging agent that is targeting tau, or if you have a drug that's targeting tau, I like to tell people who I collaborate with and I'm working with, if it doesn't affect our patients, there's really no point going on to this more complex pattern that is Alzheimer's disease. That it can be very instructive to look first at a very pure tauopathy in younger people, which is FTD. So those are kind of reasons why I think it's good to have non-Alzheimer dementias and FTDs involved also in along with these Alzheimer programs. And the particular program that I've been benefiting from is a joint program between ADDF and the Association for Frontotemporal Degeneration. So moving on to the specific program, so I'm a clinician and clinical researcher, and one of my interests is I'm very interested in developing disease, treatments that will modify the disease or slow the progression of the disease, but I'm also very interested in treatments that make people's lives better right away. So my patients with frontotemporal dementia often have very extreme behavioral changes, behavioral symptoms. Patients, as I'm sure many of, if not most of you know, patients with Alzheimer's disease often will also very frequently have behavioral symptoms, which can be very difficult to treat, very hard to make it much harder for the families to take care of the patients. And we've done some research which shows that the presence of agitation and these other behavioral symptoms increases the chance that someone won't be able to be cared for at home, increases caregiver depressive symptoms and caregiver burden, greatly increased costs of care. So we're also very interested in developing treatments to reduce behavioral symptoms, both in frontotemporal dementia and in Alzheimer's disease. So as part of that, and as part of the repurposing strategy that you've heard from other participants up here too, we became very interested in lithium. So lithium is a very old drug. You could even call it not a drug, it's really a salt, which has a long history of use in bipolar disorder. So people who are manic and actually bipolar depressed as well can benefit from taking lithium. It is interesting from a biochemical point of view because it inhibits an important step in the process of developing one of these dementias, which is what we call tau phosphorylation. So the tau molecule gets altered, and lithium inhibits that process. At the same time, there had been case reports of it improving agitation. this was actually of enough interest that NIH funded a trial several years ago. But what they did is they used lithium at the doses we give it to bipolar patients, and it wasn't tolerated. So the patients were falling, they were having other symptoms. We kind of went back, looked through the literature, and found that at a lower dose, lithium could still have behavioral effects and could still have biochemical effects. And so we then designed a trial, and it's actually going on. I think this is the first trial I'm ever aware of going on, actually both in FTD, funded by ADDF and AFTD, and in Alzheimer's patients, the identical trial using low-dose lithium, with the purpose of reducing agitation, other behavioral symptoms to make these patients easier to manage. At the same time, we're very interested in, is this disease modifying? But we feel that it will be, this particular trial is been designed to not address that question, to address the question of symptomatic improvement. At the same time, we feel it'll be a lot easier to determine if this is disease modifying, if it helps patients, if patients do better on it. So that's the purpose of our trial. We're very grateful to all of the three agencies, ADDF, AFTD, and NIH, which have funded this research program, and very grateful to speak with you today.
Thank you. So you've heard about 4 programs today, all clinical. We've seen 5,000 ideas for new drugs for Alzheimer's disease since about 1998, since we started the foundation. We've funded 500 programs in 18 countries. Currently, it's a sign of progress in the field, which is very exciting. We measure success by how these programs that start out in chemistry and in biology get tested in animals, have to go through a regulatory process to be approved for use in humans, and then this phase 1, 2, 3 FDA process to ultimately get approved. Estimated to cost, in the aggregate, about $2 billion, but most of that cost is loaded towards these phase 3 trials, that each of which cost $300 to $400 million. So a lot of innovation can come not only from the preclinical science that we fund, the chemistry, the creation of new chemicals for the treatment of Alzheimer's disease, but the kind of exploratory and pilot and high-risk kind of trials that you heard about today. Currently, there are over 100 clinical trials going on in Alzheimer's disease. That seems like a lot, and for someone who's been in the field like myself over 35 years, it is a lot. We've made great progress, but there's 2 caveats of it. One is that right now there's 3,000 clinical trials going on in cancer. So we have a long way to go to catch up and get that kind of money into clinical work and the experience of how we do clinical trials to move this forward. At the same time, I'm proud to say that out of those 110 or so clinical trials, ADDF has contributed in one way or another to advancing those trials in over 20% of them, which in my mind is a huge number for a relatively modest foundation. And I would say also to refine that number, that about 55% or more of those 100 or so clinical trials are directed to one target, which is amyloid, which may or may not work. We're going to hear something really exciting, or hopefully exciting, in the clinical trials meeting that we're having in the International Clinical Trials meeting in December. And you might get some news around December 7th or December 8th from San Diego about the success of whether this anti-amyloid strategy works. But we're going to need other drugs. We're going to need drugs like you hear from the panel today. We're going to need, just like in hypertension, patients are on 3-4 different classes of drugs. Patients with diabetes are on 3-4 classes of drugs to control their disease. Same thing in cancer. And the kind of drugs that are being developed here, I think, will contribute to those novel agents. So let me turn it over to you now, and we'd be happy to take any questions that you might have in any category of Alzheimer's. Yes, please.
Sorry. Could you tell me 2 questions I have? What causes overactivity in the brain? And how do you check if you have a Tau mutation?
Okay, well, why don't I turn it over to the experts. First one, Michela, you want to talk about what causes the overactivity?
Absolutely. So in the animal models that are in the background to the observation in amnestic MCI patients, there's actually a tendency for some overactivity of a very subtle nature to happen in aging, both in animals, and then if you use this high technology, you actually see that in people too, who are elderly. And it's there with some of the sort of mild problems in memory that happen at older ages. The thing is that in many of the animal aging models, and in many people, that doesn't take off and get worse and worse and worse. Actually, the pathophysiology of Alzheimer's takes that overactivity can drive the production of some of the molecules that are pathological, and those molecules add to the overactivity. So there's sort of a positive feedback. And then that overactivity will drive the spread of the tau pathology. So the elements of aging, the amyloid limb of Alzheimer's pathology, and of tau pathology sort of come together around this phenomena of discontrol of neural activity. Okay. And I'm not sure I've given you an adequate explanation. What the actual basis of it is that there's an incredible importance for networks of inhibitory neurons in the brain. And in psychiatric disorders, as well as in these neurodegenerative disorders, there is a sort of emerging realization that inhibitory networks may be failing. Some evidence in autism that may be the case neurodevelopmentally. So, so there are vulnerable populations, it's getting out of whack, and you need to get it back in balance to kind of control this pathology.
Thank you, Michela. Thank you. And yeah, go ahead, Sandra.
Can I comment on that, and you can then talk about the mutations? Because actually in Toronto, the Toronto Dementia Research Alliance, we're ending up collaborating with Michela and the Aging Bio study, but we're doing, hopefully with some funding from the Western Brain Institute, a sub-study which is going to be looking at the mechanisms of that hyperexcitability. It's thought pathologically that it may be the little polypeptide forms, they're called oligomers of amyloid, that are exciting the synapses too much and are making them start to sort of self-destruct. And the particular anti-epileptic, levetiracetam, quells the synaptic hyperexcitability. It's selective for the synapse. It's a unique, actually there's now another sister of it that's being developed, but it was sort of unique for many years as acting in that way. And so we are going to be doing functional MRI, some of the things that Michela had the luxury to do, and she was mainly a researcher. Now she's a clinical trialist, and so you have to keep things really simple. But we're going to have a sub-study in Toronto where we're looking at exactly how the hippocampus is activating when MCI patients are performing memory tasks with and without the drug before and after. Yeah. We're looking at electrical activity through something called magnetoencephalography. And we also may be looking directly at the tau protein and its presence in the hippocampus at the time people start into the process. So we hope to be doing studies, we call them mechanistic studies, to understand how to better answer that question.
Yeah, and actually, the Hope for MCI trial, the sub-study is Hopkins and the Toronto group together with a harmonized protocol. That's more science, but the FDA, tests for patients, prescribe it. That is a very large global study.
So let me ask Dr. Huey, can you test for tau mutations in the clinic in your practice?
Yeah, you can. I mean, so you're asking about tau mutations. Unfortunately, they're quite rare. And they're what we call highly penetrant, which means that if people have tau mutations, they will generally get frontotemporal dementia. So that families which have a tau mutation, half the people in the family get frontotemporal dementia in middle age usually. So usually we can see from the history whether it's worth pursuing testing for it. But then we can test for known mutations, and there are about 30 to 40 known mutations.
Yeah, I would say if we see a patient who is in their 50s, for example, or in their 40s, or maybe in the early 60s, that's unusual for Alzheimer's disease, and more often the second most common cause of dementia might be FTD. And so those would be the population, especially if there's a family history who we might do the test on, but it's relatively rare. But it is clinically possible just to write a prescription. Any other questions that we have? Yes, ma'am. Carolyn, okay. Sorry.
I was just going to follow up on the last question because it seems like both Dr. Huey and Dr. Gallagher, both of you are working on inhibitory systems that affect tau. Yeah. And is there a common mechanism maybe in slowing things down that keeps the tau protein from spreading?
The localization of the tau pathology in Alzheimer's early on is different than in FTD. And there are also differences in the details of how that molecule becomes distorted. So we really don't know whether mechanistically it's the same. And I don't know, FTD spreads, but not.
That's a great question.
It is.
You know, I could use you on a review panel. We just put in a grant application for this, that exact question, because one of the things that's very interesting is with how you get this diverse spread. So even within the grant we put in to NIH was looking within families with tau mutations, you get this huge range of phenotypes where you get people who have behavioral FTD, you have people who have motor disease, which we call corticobasal syndrome or progressive supranuclear palsy, with the exact same mutation in the exact same family. So and this actually brings up the epigenetics and issues like that. There's things we don't understand going on of why tau is behind so many of these illnesses but goes to such different places.
And interestingly, the familial genetics, there aren't variants that affect the memory system quite the same way Alzheimer's does. But in experimental models, you can stick that gene into the neurons where degeneration starts to occur, and it does behave along the connectional system the way that Alzheimer's does.
Okay.
So it's,
I want to move on to giving other people some point for question. Go ahead, please.
Yes. Have you found any connection between nicotine poisoning and the development of Alzheimer's? I'm thinking particularly of heavy smokers whose autopsies show perfectly clear lungs. Could the heavy smoking have triggered an Alzheimer's?
Let me answer that. There's sort of two sides to the nicotine question. One is, does smoking per se cause or contribute to Alzheimer's disease? And there's a lot of evidence that smoking is definitely bad for your brain, and if you're a heavy smoker, it's bad for your brain, basically. And when I see patients who are heavy smokers who have memory problems, for example, one of the first things I'll do is tell them to stop smoking. There's many ways that smoking is bad for your brain. Vascular problems, toxins that get up there that are neurotoxic, and so on. Nicotine per se, one of the chemicals in the brain, it's kind of a double-edged sword. We're actually funding a clinical trial of nicotine right now. We recently funded it for treatment of MCI, of mild cognitive impairment. The reason is that nicotine is a chemical that's involved in the cholinergic system, which is part of the chemical system of the brain that forms new memories. And current drugs on the market, like Aricept and others that are the main class of agents, function in the same system, slightly differently, but function in the same system. And there's some evidence that in various neurodegenerative diseases where there's cognitive impairment, nicotine can be beneficial. So there's the nicotine per se in cigarettes in the right dose and delivered in the right way actually might be a treatment for Alzheimer's disease because of this cholinergic chemical problem. But smoking is really bad for your brain. And if you're smoking and you want to prevent or delay the onset of dementia, you should stop right away. Okay, let's see. Oh, do we have the microphone over there? Yes, please.
So, to what length of time can people with MCI just stay with MCI, and do some never develop any more serious diseases? And how does modafinil and that type of drug help mild cognitive impairment?
Modafinil, is that what you said? Yeah. Let me just give a broader picture of MCI. So MCI is a clinical diagnosis. So what that means is that we see people, and basically for most people, Ted mentioned, Dr. Huey mentioned amnestic MCI, and I think Michela mentioned it. That means that these are people that just have memory problems. They have memory problems greater than what we would expect with normal aging, but they don't have other kinds of cognitive impairments that define dementia. They don't have trouble tying their shoes. They don't have trouble with abstract thinking. They don't have trouble with planning and so on. So that they don't have functional disability. So these are people that have memory problems. So if you take 100 people that came into a doctor's office with the diagnosis of MCI, actually only half of them would have Alzheimer's disease or some form of dementia. And about half of them actually we might be able to find a reason why they have MCI. Maybe they're on too many drugs. Maybe they're taking too many sleeping pills. Maybe they, doing other things. Maybe they're very depressed and that sort of thing. And some of them might just have a vascular form of what we call vascular cognitive impairment, which in many people is very slowly progressive. And that's where this new amyloid test, this PET scan, comes in really well. I think it's going to be the main use of the amyloid PET scan that our foundation helped to develop. Because these people are very hard, it's very hard to know what their future is going to be. The 50% of people who have MCI, severe, serious memory problems, and a positive amyloid scan or positive spinal fluid, they'll become demented in 3 to 5 years. 50% of those people will become demented. We know that those people have a high risk of developing full-blown Alzheimer's disease clinically and dementia in 5 years. But 50% of those people or so may not progress. And one way to figure that out would be to do an amyloid scan, and we're having other new scanning techniques coming up. So the diagnosis is more of an umbrella, and we can subpopulate that. Modafinil is not used for Alzheimer's, and there have been 1 or 2 studies of modafinil, which is a stimulant, and we haven't found any use for it.
Can I just mention another sleep disorder, though, that can commonly cause a problem that mimics MCI is obstructive sleep apnea, people who snore and have apnea. That's one of the differential diagnoses sometimes of this problem.
Can we get some questions on this side over here? Yes, sir.
Is there any relationship between a history of concussions and Alzheimer's?
Yeah. I can probably take that one pretty quickly. So we're actually working on concussions. We're funding a study at Mount Sinai using this amyloid imaging and tau imaging. So now there's new scans that will probably come to market, I expect, in the next few years that can actually look at tau. And the difference between amyloid and tau scanning on PET is that amyloid is somewhat diagnostic. Tau is, you can see these neurons that are dying. Those are the dying neurons. And they correlate much better with cognitive dysfunction. Concussions lead to more of a tauopathy, but we do know that some of the concussions also have amyloid deposition. And there's a fair amount of data that multiple concussions are a risk factor for Alzheimer's disease in late life, although that's not set in stone. So concussion is this chronic traumatic encephalopathy that the professional football players and others suffer, and maybe even soccer players that are heading thousands of times a season. They're not getting big concussions, but they're getting multiple small concussions. Those people have cognitive impairment 6 months after the concussion. We know that. The problem is whether they completely heal or whether they go on to this chronic problem, whether it becomes chronic traumatic encephalopathy in their 30s and 40s and 50s, or whether ultimately that's a risk factor for Alzheimer's disease. We really don't know. I think we have, unfortunately, just time for one more question, and that's always a hard thing to do.
May I
Yeah, please do.
A small detail on concussion. I mean, we know, for instance, that in concussion, in traumatic brain injury, there is one biomarker who is a neuroinflammatory biomarker, it's 189, which is highly overexpressed, which has been also found to be overexpressed in Alzheimer's disease, and which has been also found to be overexpressed in postoperative cognitive dysfunction. So the neuroinflammatory role is probably playing a role on those diseases, and might be a mutual link on that.
Yeah. So you've heard about many risk factors now. Sleep apnea was mentioned, smoking, concussions, and so on. So we have many risk factors that we've identified. Last question over here.
Hi, I'm more interested in a possible prevention for my children who are now close to 50. My husband, Dr. Graham, the slowing of the beginning of Alzheimer's did not really apply to him. He went from doing active research at Columbia in the NCI within 4 months to full-blown Alzheimer's. So I am hoping that any of you know a lecture we had at the Lotus Club, they were hoping for a vaccine within 20 years. And I'm hoping that you also have some encouraging notice, because if my children get Alzheimer's, which impairs them as quickly as it did my husband, delaying it is going to do no good. I mean, he literally went from getting a big grant for Columbia Cancer Center, he'd done the work, obviously, the 2 years before, to my daughter, who's also an oncologist, having to go up and tell the head of the cancer center he had to be retired. And that was within a 4-month period. I noticed something, his accountant noticed something, and then 2 months later, full-blown?
So let me just, you raised two things that I think is worth mentioning, prevention and the vaccine. And the vaccines, we often hear a lot about vaccines now. What we're really referring to are monoclonal antibodies. They're biologicals that are injected. They go into your brain, they bind the amyloid, and they're trying to remove the amyloid from the brain that we think is neurotoxic. So these are referred to as vaccines, and they are, in a sense, vaccines. And we're very excited, as I mentioned, in early December, we're going to hear a report from Eli Lilly about whether a vaccine that they're using is going to work or not. The vaccine, with these amyloid scans, the real advance has been that we can, maybe in your daughters, for example, they can get the amyloid PET scan. And the amyloid PET scan that our foundation helped to develop has been a breakthrough because we can identify people before they become symptomatic. The vaccine has shown that the Alzheimer's starts 20, 30 years before people become even MCI, before they even start to have memory problems. So we can do that now, and now that's enabled is not only to test people that are concerned, like your children, and maybe they can get a genetic test like ApoE to see if they're at higher risk. Because now there are prevention trials with the vaccine. So what's happening, which is amazing, is going on something called the A4 trial and others, where people who are screened in the community that are normal, who have a positive scan, so they have nothing wrong with them, but they have a positive scan, so they have the disease, but they haven't been affected, are going to be treated to see if we can prevent symptoms. And these are the first prevention trials that are going on in our field. And so that's very exciting that we're doing. Everybody would get treated as they age, and we would erase dementia from the world. And with that, I want to introduce I want to introduce Randall Sandler, who is the vice chairman of our board and a great supporter, and we're very thankful to Randall for everything he does for ADDF.
Thank you, Howard. That was a very optimistic note to hit in on. I want you to please join me in our thanking Dr. Phil and our esteemed panel for the program today and for all their insights on a lot of the research studies that we're funding at the ADDF. So, thank you. I know I'm not alone in saying that all the work that you do means so much to us and our families, and it's really yeoman's work. I also want to thank all of you for attending our program and for all of your support. I hope it was an informative and helpful program, and it gives you a window into the types of research that we're funding, especially the innovative research that we're funding at the ADDF. There are lots of programs, and as Howard said, it's pretty astounding that over 20% of all the clinical trials in Alzheimer's, that the ADDF really has its fingerprints on those trials. As Rondell noted, this is a very exciting time in drug discovery for Alzheimer's and dementia. Thanks to you, there is significant hope on the horizon. And with that, I invite you to start to make your way to the ballroom for our lunch and award ceremony. Thank you.